Looking at life in your soil and just having a reflection on how soil biology works and how that might feed through to farming practice. I guess the most important thing for me to say in the first instance is just that soil is great, very exciting. You all need to actually go home and get a spade and dig holes, because that's the only way you can see what's actually happening in soil and what's going on. We need to also put together the physics, the chemistry and the biology, and we're going to focus particularly today, certainly this first session, on physics and biology, and we're going to come back to nutrition and some of the chemical measurements in soil this afternoon. But you can't separate those things out. In terms of managing healthy soils, you have to understand and work with the physical skeleton that your soil gives you, work within that, understand what surfaces you have, how reactive they are, how they're holding and manipulating the inputs that you put in. What that means in terms of how that soil is able to hold on to organic matter, also strongly related to the active surface area that the soil particles give you. And then how that feeds through to how the soil's structured, so the space that exists between the particles of soil, and the pores become the place that all sorts of interactions happen in soil. They're kind of like the motorways and the road networks in which things happen. Things move and are changed and exchanged in that pore space, including water, and that's critically important as we think about managing soils for access for livestock in terms of improving trafficability and reducing damage. But also because these pores provide the place where plant roots need to operate, which is critical for production, and also where the soil biology largely operates. And most of that soil biology is aquatic. It's been inherited from ponds and streams and rivers. It continues to live within the water films in the pores. Some of the large soil fauna can move between and across water films. They're kind of like expert mountain climbers getting over the hard bits between the water films. But the smallest organisms live and swim and operate completely underwater the whole time. That doesn't mean they don't need air. That air is diffused through the water films, through the pores. And our pores are going to contain either water or air. Kind of how it works. So we've got a skeleton, non-living, against which we integrate and hold soil organic matter that's been drawn from photosynthesis through plant roots over 10,000 years in most of the UK as those soils have been forming. And then against that we see the operation of the soil organisms and how they're taking things forward. And if we just look at soil life as a whole, usually there's someone who can read these numbers and spoils my guessing game. But here we have 500 grams of soil in a jar, taken from a grass clover ley. Nothing particularly special about it. We did it for a magazine article in 2006, which is hence the date. And we did the numbers. So we've got some numbers of bacteria and protozoa and nematodes and fungi. And the simple answer is there's lots of it. Those numbers are pretty big. There are 12 noughts on this number of bacteria that are in that soil. That's an awful lot of them. There are 5 million single-celled protozoan organisms in that jar of soil. That's an awful lot of them. And the one that blows my mind every time I see it, and I still can't get my head around it really, is there are 10 kilometres of fungal hyphae. So about 6 miles of fungal hyphae in that jar. It's like, what? How does that happen? I know how it happens because they're very small, so they can fold it, but every time, how can 6 miles of anything fit inside that jar? So we've got a huge diversity, complexity of soil life happening here. Hugely species rich, hugely diverse. When I started doing soil science back in the 1980s, we could only describe this biology by taking it out of the soil and putting it on plates or other culture media and looking in that way. And as we did that, we thought we were understanding what was going on, and we started to get a handle of quite a lot of processes using methods that had been developed and extended into the 1980s from the 1920s and so on through medical science. But of course that was also the point at which DNA extractions and CSI got very excited about using barcoding. And as we started to use that in soil, we discovered that what we saw by plating up soil organisms was less than 5% of the diversity of the organisms there. So when I started soil science, there was 95% of this soil life. We had no clue what it was or how to call it or even what it was doing. And we're beginning to put that information together now and draw it together in a way that we can use that information to change and manipulate our management practices to benefit or work with those soil organisms. Importantly, these soil organisms don't uniformly operate across the whole matrix. I guess I talked about motorways before. If we flew across the UK at night, looked out the window, if it's not cloudy, we would see the UK laid beneath us, and we wouldn't see street lights in a evenly distributed across the UK kind of way. What we see is clusters of lights in towns or villages, ribbons joining those villages and towns together. And the same would happen if we could do the same sort of thing with soil life, and we kind of can. I haven't sneaked a photo in, but we can use photoluminescence to have a look at soil organisms, and we find that the patterns we see from our planes at night and we look at soil in that way aren't very different. So what we get is clusters of organisms in interesting places where stuff's happening, where there's life, usually where there's energy feeding into the soil through plant roots or recent inputs of organic matter. That said, the bit of the soil that looks dark in that situation isn't necessarily dead. It may just be waiting for something interesting to happen. Across much of the UK countryside, there will be people sitting in their houses in an evening watching CSI or some other interesting box set on the telly, and you won't be able to tell that they're doing anything very interesting because their lights won't be shining. It's only when someone has a party in the village that suddenly the village comes to life. You know those village parties, I'm sure, you know. Yep. And so the same sort of thing is happening in the soil. We've got those organisms waiting for something interesting to happen. They will respond to an invitation, usually an invitation from a plant root, the exudation of a signalling compound that says, hey look, I'm here, there's something interesting happening. Or a sugar, a simple sugar compound released through photosynthesis, will suddenly enliven and bring to life something you thought wasn't doing anything at all. And we certainly have evidence from soils that have been buried, for example, through landslips in the post-glacial period even, so 7, 8, 9,000 years ago. Things that were topsoil then that have now been buried under 20 metres of rockfall. When we go back in and take those soils and subject them to a treatment like giving them a bit of sugar, they'll actually respond and start breathing because the organisms have been able to sit in a dormant state, some of them, not all of them, for that sort of thousands of year period. The guys who operate in soil often operate in cascade, so the enzyme of one will break down something else, and normally the processes that are happening are external to the cells of the organisms, particularly the smallest organisms. So what we have is a network of enzymes being released into the soil pore water, which then make things happen. And as a consequence, we have a lot of shared working, a bit like production lines in car factories, though with active organisms rather than robots these days. So we've got the specialists too, though, things like nitrifiers, where those have particular adaptation, enzymatic adaptation, to use, for example, in the case of nitrifiers, the ammonium fertilizer that's added to generate the energy they need, and they do that by turning ammonium into nitrate. If we want to know how much biology our soil could hold, we could do something quite complicated, like make a diagram that makes everybody very confused. But you know what? This is a simplification. This takes the idea of something from, I think arable farmers are very comfortable with the idea of potential and attainable yields, and it's true now also in terms of thinking about grass varieties, and express that in terms of looking at, and this is particularly for the smallest organisms, the soil microbial biomass. Because, well, we know that not every soil will be able to contain the same amount of biomass. There just isn't the same amount of space in different soils. So soil texture, depth, parent material, set the skeleton. A sandy soil has much less surface area, much less space, therefore, for the organisms to operate than a clay soil. So we're going to have, just by those limiting factors, the skeleton, some constraints. And then there's food. If there isn't much to eat, there won't be a very big active population. So the defining factors that set the attainable level of these organisms is to do with how much we fed them. This sounds very familiar to livestock farmers because this is about both space and housing and food. And those two things are critically important for any livestock, whether we're talking about livestock below ground or livestock above ground. So if we're managing the livestock below ground, we need to have a thought to those two things too. What are we doing with the space below ground? And the next guys who are going to speak after me are going to follow that out beautifully. And also, how are we feeding these organisms? What's coming into the system? What kind of inputs? What kind of plants particularly are growing? But also, what are we doing in terms of managing those things? And we can be quite sneaky, it has to be said, in terms of soil management, because we can bring in net primary production from other places. The materials that we might have available to us, and arable farmers tend to use them more than grassland farmers, like green waste composts, but no reason they shouldn't or couldn't be used in livestock systems, again, to usually improve soil condition and structure. But those things are a way of us bringing somebody else's net primary productivity, someone else's photosynthesis, into our piece of land, which means we can then potentially support organisms that our own net primary production on its own couldn't do. And of course, within a livestock system, we're quite good at moving our net primary production around the farm in things like manures and slurries. The production from one field comes actually into another. And we often think about that in terms of nutrients and nutrient budgets, but we're doing it too with the carbon and energy that we've trapped, so that we actually have quite a lot of tools, management opportunities, to control and look after our biomass. But perhaps what we're interested in is not the total amount of these organisms, but what they're doing. So let me just tell you, they do pretty much everything. There's kind of nothing that this biology won't get involved in if you just leave it to do it. Particularly, the soil biology drives the decomposition of organic matter. Well, of course it does. That's where it gets its food from. Its energy comes from the decomposition of the fresh and older organic materials in the soil as they're broken down. But it will also, well, work on anything you put in. So any chemical that's added to soil, after a while, and it does sometimes take a while, the soil organisms will begin to break it down. Even engine oil, the soil organisms, give it a while, they'll work out a way to use their enzymatic capacity to start to break that material down. And it's one of the reasons that biodegradation is A, a big problem to chemical manufacturers, and B, a great route for exploiting in terms of thinking about waste treatment as we go forward. But it's actually biology that also has a key role in driving soil aggregation. Yeah, machines are a little bit important as well, but actually it's the biology that makes soil aggregates more resilient in terms of giving them short-term structural resilience, holding and pushing apart soil particles in a way that creates medium-sized mesopores in a way that tillage can't. Directly, these organisms can affect nutrient uptake by plants. I'll have a quick tour of the plant-associated organisms. We've got degradation, I've already talked about. Critically important, of course, in terms of plant disease. And for grassland farmers, the awareness of the importance of managing soil pests, especially for reseeds, and thinking about how those things go forward is critically part of soil health and how we keep a balanced set of ecosystems working there. So to use biological control opportunities in relation to plant pathogens too. So bacterial and fungal allies, just a couple of pictures. These ones guys are quite hard to show you pictures of, so it's best to show you them in association with roots. You will be familiar with these. The easiest ones to take pictures of are beans because they're biggest nodules, which is what that plant is rather than a clover. The nodules here are the result of the close symbiosis between the plant and the organism in the soil. The plant sent out a signal, the organism turns up to the party, makes a cross-cell wall relationship. This isn't just any old relationship, this is a mutual dependency. This is the kind of really full-on relationship that, you know, excludes everybody else. You know how sometimes you walk into those rooms? Yep, excludes everybody else from the conversation. So here we have the plant making a space for the organisms to work most effectively. The plant creates a special kind of hemoglobin which keeps oxygen out because it's really important for the microorganisms, the rhizobia here, that they're in an oxygen-free environment because that allows the processes then to happen. Sugar provided directly to these organisms through the flow through the plant, and then the organisms will respond, thanks, by trapping and fixing nitrogen from the atmosphere. Really close relationship of benefit to both plants and organisms and of side benefit to other plants that are hanging around in the vicinity. So it's why clover within a sward will benefit the grass too. Quite temperature dependent, we're starting to understand the relationship between specific rhizobia and specific plants more clearly. and an awful lot of work and an awful lot of conversations that we could have about do we need to or don't we need to inoculate and how might we make that work. But perhaps more interesting, and the thing we didn't used to know so much about, a soil mycorrhiza, which is a fungi. Now this kind of relationship isn't a symbiosis, it's much more like a mutual parasitism. I do something for you, you do something for me, we both rub along together, sometimes it's good, sometimes it's not so good, but somehow we work it out. So a lot more like normal kind of relationships, really. The soil fungi here, again, invades the plant cell. So this is the cell wall here. The fungus operating both inside and outside the cell, extends its hyphae potentially miles, but certainly meters, into the soil around, and that is connected directly to the inside of the cell through the fungal hyphae. Inside the cell, the fungus proliferates and makes what looks like a tree structure. These are called arbuscular mycorrhizae because of that tree structure that we can see under the microscope. In that tree structure, they absorb the nutrients from the plant, which the plant gives them because it sees that it can gain benefit from the nutrients, water, and other things that the arbuscular mycorrhizal hyphae are able to bring to the plant. So if in this exchange area, the plant gives sugar, of which it has plenty through photosynthesis, but takes out phosphorus and micronutrients, the concentration here of phosphorus and micronutrients within the fungus will be reduced, and consequently they will flow along the fungal hyphae inside the cell wall system, which stops them being held up or restricted in the way that phosphorus would normally be as it tries to move through the soil to a plant. So we can see quite big improvements in phosphorus nutrition in low P soils. It might be one of the reasons why grassland farmers might have soils with P indices of 1 but no problem with phosphorus nutrition. The kind of biological driving factors that might offset the lack of chemistry or the wrong sort of chemistry, the fixation of phosphorus within soils. Decomposition happens because of a complex interaction between organisms. It's not enough just to have bacteria and fungi. Those bacteria and fungi need to be eaten by somebody else. Bacteria and fungi are pretty conservative. The stuff they break down as they chew over plant residues or they let their enzymes chew over plant residues tends to be conserved quite tightly within the fungi and bacteria. They're not that generous in the sharing that they do with nutrients through mineralization. But as they get consumed by higher organisms in the food chain, actually these guys aren't so good at holding on to nutrients. In fact, they're just greedy. They eat more than they need in general. Their carbon to nitrogen, carbon to phosphorus ratio means that these guys tend to release nutrients. So it's at this point in the food chain that we tend to see the release of nutrients. So if we do something to soils that gets rid of all these, and I guess the kind of example that might are things like high-end nematicides, for example, not commonly used in grassland systems, but we will see that sometimes in horticultural systems where there's been an intensive use of nematicides over a long period, that actually there'll be bacteria and fungi there, but not so much food chain up the way, which means there isn't a big release of nutrients. The big organisms are important though because they chew over these residues in this organic matter to break it down, so that they become important at both levels in the food chain. What we've got here is a whole complex interaction. It was suggested that this might make quite a good computer game, known as Soil Wars perhaps, where something eats something and we work out. I think it's probably worth a go if anyone in the room fancies a gaming opportunity. The thing you probably want to notice though is the earthworms are over there somewhere, because they're sandal-wearing vegetarians. They're not really interested in being part of this war. They're pacifists, they're vegetarians, they don't really care what's happening over here. The earthworms do that because they have their own inbuilt, in-gut microflora like we do. So actually they don't need to work with these soil organisms to digest and get the organic matter they need. They have that sort of built in. So therefore what they do is respond directly to particularly the input of fresh plant residues and the existence of roots in the soil through the year. So a system that has fresh residues, and when I say fresh residues, grassroots count, going in all through the year will tend to have higher earthworm numbers. The other thing that earthworms really dislike, any big organisms dislike, is someone who comes along and turns their house upside down on a regular basis using a tillage implement. They're not best impressed by that, so the population of earthworms in a tilled soil, for example, in rotational rather than permanent pastures, will generally tend to be lower. And you'll see if you dig those holes, which you're all rushing home to do, I know, you'll see much bigger earthworms in things like permanent pasture than you will in rotational lanes. And if you have fields that are out there in permanent arable cultivation, they will tend to have an even more juvenile population, a population that's required just because of the tillage to keep on updating itself and running through. And there they are. Lots of different kinds of earthworms, all with slightly different functions, particularly different life cycles, and that means they affect the soil at different levels. I think I've got the link in, but the Healthy Grassland Soils page, which you can access through Dairy Co's website, and I think I've put the link in properly in a minute, gives you access to the resources that if you want, you can start not just counting earthworms, but understanding which ones we've got and what they're up to in soil. And that can be important, particularly if you're in a situation where you might have damaged, and these guys will come back to that later, pore space, because they're the anesic earthworms, the lobworms, are the ones who are best at rebuilding structure quickly. Those soil organisms that I've described are not there for you. They're not really bothered about you. They're there to do stuff for themselves. So what they're doing is making the environment work best for them, whether that's about modifying soil structure, breaking down organic matter, or liaising with plant roots in a range of ways. And just to show you some of that complexity, and there's a whole set of these entertaining diagrams that kept me busy for at least 6 months, a couple of years ago. We've got the decomposition process that I showed you a minute ago as the engine of what's happening. This is soil structure. So it's decomposition in soil that releases polysaccharides and glycoproteins that then act as the glues and binders that put the sand and silt and clay domains together and link the organic matter. There's also, of course, some direct fecal pellets, actually really quite important in terms of holding soil aggregates together. The roots themselves add glycoproteins and polysaccharides, but also act as a way of holding those microaggregates together with that fungal hyphae, that miles of stuff there too. And so we get aggregates and microaggregates, and in a biologically driven soil structure situation, we will see rounded, open aggregates that are quite porous. In a driven by tillage system, go to an arable neighbour and have a look, you'll see much squarer, sharper aggregates because the biological processes are less common. And then those pore spaces are then manipulated by what the jargon might call ecosystem engineers, the guys who go through and make motorways, earthworms, or A-roads, the Enchytraeids, Calembula and mites, literally making pores through the soil as they go. Generally, why do we care about this? Well, because the data suggests that if we can increase the microbial biomass in a particular soil, we'll actually increase the amount of nutrients we're able to supply through biological processes. So we might not just be interested in them for themselves, we might actually want to make these guys work for us in the system. This is data actually from arable systems because we have better data at the moment on the direct link between nutrient supply and soil microbial biomass in those systems. There it is, there's the link. So managing soil life means understanding it, means recognising it's there, means husbanding it, perhaps in the same way as you might husband your livestock. And there's quite a lot of, I would say they're good because I was involved in putting them together, and I will say they're interesting for the same reason. But there's information there that takes us through how different practices on farm across a spectrum might affect this soil life, what the role of grazing or herbicides, etc. is in that relationship. Also some background information, the kind of stuff I've just trawled through for you. But the overall messages are actually incredibly simple. I don't know if anyone saw Kevin Costner, it was on the telly not long ago. I was very excited. The key here is not that we need to introduce new soil life that's not there. What we need to do is create the place for the soil life that we have, some of which will be dormant, to operate most effectively. So what we're looking at here, if we want messages to take home, are about system-orientated approaches. And in that context, they're about feeding the livestock below ground, both through direct organic matter inputs, and most grassland systems are already doing that well, but what they could do is do the second, which is increase the diversity of plants that are operating in the system, provide a more varied diet. It's starting to sound like a human health talk, and it is. We need to have the right diet of the right sort to give a diverse and a resilient population. And at the same time, particularly around reseeds, grassland farmers might want to think about tillage intensity and what that might be doing to the biological systems in the soil.